Can you folks see and hear me?
Yes. Yeah.
Excellent.
I hear you now. Excellent. Thank you, Mia. I'm going to introduce you and we can get started. So our last presenter is Mia Akbar. Mia is a first-year MSc candidate studying biology at Queen's University. Their current research uses quantitative genetic methods to investigate rapid evolution of the invasive plant purple loosestrife in response to biotic and abiotic stressors such as climate and herbivory. In this talk, Mia will discuss the research findings regarding the interaction between climate adaptation and biological control agents for purple loosestrife. And with that, I'll give it to you. Thank you.
Thanks, Vicki. I have to say I'm excited to be presenting after those last two presentations because I think that some of the research I'm going to be presenting today actually fills in some of those gaps that we see where like the species distribution models like have some variation, and I show some of like the potential biotic and like locally adaptive factors that play into what makes it so that a species can be successful or non-successful in introduced environments. So with that, like Vicki said, my name is Mia, and I'm joining you today from Kingston, Ontario, which is situated on the traditional lands of the Haudenosaunee and Anishinaabe. I'm very grateful to live and learn and conduct science here as an uninvited guest. And I'm currently working on my master's, like Vicki said, at Queen's University under the supervision of Dr. Rob Colautti. And today I'm going to be presenting some of the past, current, and future research of our research group. Oh, there we go. So to begin, I'd like to introduce you to this very pretty but problematic plant that I'm sure many of you folks have seen before. So purple loosestrife is a wetland perennial plant that's often found in ditches and wet meadows, marshes, and alongside lakes. Disturbance often germinates its seeds, which is why it's commonly found in human disturbed areas. And when purple loosestrife initiates its flowering, it shifts resources away from this vegetative growth that we see here towards buds, fruits, and flowers, and then subsequently fruits. And then once it does eventually fruit and produce seeds, it can produce, one plant can produce upwards of 2.7 million seeds per season. So that's definitely part of the reason why it's become such a prolific invader. It's initially native to Europe and Asia, and it was introduced to North America in the 19th century or earlier. That's just what we have records for. It underwent multiple introductions through ship ballasts, imported livestock, seed for gardens, and beekeeping. And it has spread across the entirety of, almost the entirety of North America. Something interesting to note about this plant is that it does kind of follow that lag response time that we saw in those modeling papers, where it was introduced in the 1800s at least, but it has only undergone rapid spread in the last 50 to 70 years. And as everyone attending this conference is well aware, the effects of invasive species can be incredibly taxing on both biodiversity and the economy, and purple loosestrife is certainly no exception. It's squeezed out many native plant species and has been the subject of many expensive management programs. None more notably than its biological control program. So biocontrol is the use of the species' natural enemies, such as herbivores, pests, pathogens, and parasites to reduce established populations and invasion. This was a program that was implemented between 1992 and 1996, where these and this weevil was introduced. However, there is huge success in these biological control programs. In some stands, it's been recorded that more than 90% of biomass has been reduced, but in others, it appears that the biocontrol has had little to no strong effect, and individuals are still able to survive and reproduce and persist despite the beetle. You can see here in this figure where we've got, percent cover on the y-axis and years after release on the x, that there's like a general trend of decrease in biomass, but there's still a lot of uncertainty. It's a pretty big standard error, and also huge variation between sites, which is a pretty big indication that there are more things affecting these plants than herbivore pressure. So as ecologists and evolutionary biologists, one of the things that our research group really wants to know is why we see this variable response to biocontrol. What factors, both biotic, like herbivory, or abiotic, like climate, influence rapid evolution of this plant? So why do we see that some of them are more resilient than others? What makes it so that they can grow across such a wide range of North America? The climate up here in Northern Ontario is very different than in the Southern United States, but yet it's everywhere. Do these things interact? And are their responses from North American purple loosestrife different than the responses of European purple loosestrife? If we're able to answer some of these questions, we may be able to provide better information for managers to use when developing a framework for the implementation of biocontrol programs, not just in purple loosestrife, but in other invasive species systems as well. So to answer some of these questions, our research group is using a highly intersectional approach that aims to attack this problem from multiple angles. We're utilizing genomics and transcriptomics and population genetics and quantitative trait analysis. To this end, we aim to contribute to purple loosestrife, the purple loosestrife model system, so that it can be used to answer important questions about ecology and evolution related to invasive species, and create empirical evidence that can inform mitigation and management efforts. So further knowledge gained here will hopefully allow us to predict also how native plants are going to respond under anthropogenic climate change. So what have we done so far? In common garden experiments that we've conducted, we see that growth and timing of reproduction is highly variable in the introduced range of purple loosestrife. What we see, plants that grow at northern latitudes stay small vegetatively and reproduce very early in the growing season. So you remember at the start of the presentation, I mentioned that at a certain point in the growing cycle, purple loosestrife will switch from that vegetative growth over to reproductive growth, and then the size that it's at when that happens is kind of the size that it stays at. So plants that flower earlier, in order to get through an entire reproductive cycle before the first frost, will stay small and produce less seeds from southern latitudes that have a longer growing season, and therefore can take a longer time to get nice and big vegetatively before flowering, and then ultimately produce more seeds. And we see that this is a pretty consistent across latitudes. Further, when we grow these plants in a common garden experiment, specific transplant experiment, where we grow northern plants in the southern environment and southern plants in the northern environment, we see that this variation is genetic, because when northern plants are grown in the south, they still flower early and aren't able to produce as many seeds as the southern plants. And when southern plants are in the northern garden, they aren't able to flower soon enough, so they don't finish their growing cycle before the first frost hits, and then they experience a huge hit in reproductive fitness. So this just is related to how many seeds they produce. In evolutionary biology, we call this phenomenon a genetic constraint, limit or a constraint to the way that this plant can respond to its environment. Because of its genetics, the southern plants can't flower early enough. And because of their genetics, the northern plants flower like quote unquote too early and aren't able to exploit the entire growing season. In this way, genetic constraint imposed by this adaptation to climate changes the way that purple loosestrife can evolve. And these genetic constraints may limit the ability of purple loosestrife to evolve in response to herbivore pressure. And this may explain some of the variability we see in these biological control programs. So some of the future work we're doing to investigate the effect of the genetic constraint and the herbivore response is growing plants from a variety of latitudes, northern Ontario, all the way down to mid-latitude in the United garden experiment in like the mid-latitude range. So this garden's located at the Queen's University Biological Station, which is just north of Kingston. So we have sample populations, like seed families from all of these latitudes, and we grow them in this common garden experiment. All in the same environment, and we experimentally manipulate the herbivory that these plants will experience. So half of them are sprayed with insecticide, and the other half subject to insect herbivory. And then I'll compare their what we call total reproductive biomass, so just the seeds they produce, growing season after growing season. And in that way, we can also capture how much of these differences are attributable to just year-to-year variation as opposed to genetic differences between plants. Further, we're taking plants that are grown in this garden and also doing some molecular genetics with them, where we're evaluating, trying to identify some defense genes. If we're able to identify a difference in these defense mechanisms at both the molecular and quantitative level between plants that flower early and plants that flower late, it seems that some plants may be able to evolve faster than other ones, and this could support the introduction of a more aggressive biocontrol to specific regions, or at the very least allow us to predict, you know, in which areas plants are going to spread next. Finally, I would just like to take a step back from the purple loosestrife system and consider other invasive plant systems and invasive species generally. One of the primary objectives of our research group is to conduct science that is applicable to real-world problems and interpretable by real-world problem solvers. The rate of invasions is steadily growing, and climate change will only exacerbate these issues. And our goal is to use the purple loosestrife model system as an evolutionary model for invasive species so that we can provide empirical evidence for the managers that will ultimately make these decisions, but also predict how native plants will respond to the myriad of novel pressures that they'll experience under anthropogenic climate change. With that, I will thank some of my colleagues and my advisors that contributed to this work over the years. The kind of work that goes into running a common garden experiment at the scale that I showed you folks is astronomical, and it's not lost on me how privileged I am to be able to take advantage of that field data for my master's project. With that, I will take some questions.
Excellent. Thank you, Mia. Is the southern northern flowering timing an epigenetic phenomenon?
So no. The short answer is no. When the plants likely came over from Europe, there was like sufficient genetic variation for some plants in like more northern areas to be selected for an early flowering time, like by natural selection, and some plants to be selected for a later flowering time. And then those plants were the ones that were able to be successful in their respective regions and then expanding stepwise in that way, if that makes sense.
And would purple loosestrife be considered a sleeper species? That is to say, a species that persists at low population levels until favorable climate conditions allow an explosion in growth?
If that's in reference to the lag time idea, that's like definitely a possibility. I think that that's might be a hypothesis for why we see like that lag time. But another idea like posed by like population geneticists is that it takes a certain amount of introductions to create enough genetic variation that we can see that rapid expansion in the first place. So like what I mean by that, if there's only like a small population and there's only like one type of like if there's only one genotype in the population, natural selection, when it acts on it, can't select for genotypes that will flower earlier or genotypes that will flower later because they're all the same. So it takes gene flow from multiple different populations, from multiple different introductions, to create enough like evolutionary material, so to speak, so that natural selection can even work on the system. So I think, like, from a population genetic evolutionary perspective, it's more likely that it's the multiple introductions, and eventually there were kind of just enough, like there was enough genetic material for natural selection to work on. I hope that answers the question. I'm not like familiar with the concept of sleeper species, so I don't know if that qualifies.
Is anyone looking at how much genetic variation or adaptation is going on in the biocontrol beetles?
No, but that's an upcoming project idea, is looking at the potential like co-evolutionary dynamics between biocontrol beetles and purple loosestrife.
Excellent.
And people are doing that in other systems. I know that that's something that's going on right now in the mountain pine beetle study system. So looking at like co-evolutionary dynamics between like lodgepole pine and mountain pine beetle. Obviously very different than, like a herbaceous perennial, but yeah.
Great. Thanks so much, Mia.
Thank you, Taylor.
Thank you. Okay.